on how molecules get across the membrane is energy needed to cross the membrane? Sometimes you need energy and sometimes you don't. And there's a simple way to tell whether or not energy is going to be needed. And for this, if you understand the concept of diffusion, that'll help you decide. So do you guys remember, what's the main concept of diffusion? What are we looking for?
High concentration gradient.
Good, a concentration gradient, right? So remember, for diffusion, we're looking to see, is there a high to low concentration? Right? And if there is, what do you know about how the molecules are going to move?
By themselves.
They'll move by themselves, right? So in diffusion, the molecules naturally go from high to low concentration. So remember, because they go by themselves, this is considered passive, or no energy is needed. So if there is no energy needed, or if it's a passive transport, that's when you're actually just following diffusion and using diffusion. So for example, in this picture, right, think of out here in blue is the outside of your cell, and then in here beige is the inside of the cell. And if you just take a look at the green molecules, where's the high concentration of the green molecule? Outside on top or inside on the bottom? Yeah, outside. So based on diffusion, where do the molecules want to go? They want to go in, right? Or they want to go down their concentration gradient. And again, that happens, no energy is needed. Kind of like lab last week, right? You dropped the purple crystal in the middle or the methylene blue in the middle. What happened naturally after you dropped it in the middle? What did it do? It automatically diffused outward, right? After you put the initial drop in, notice you didn't have to do anything. You didn't have to apply any more energy, anything extra. It just happened naturally. So again, that's passive, no energy needed. Yeah,
So
sure.
once there's an equal amount of molecules or equal.
Exactly, yeah. Remember, diffusion will go until there's no more concentration gradient. So once the molecule concentration evens out, here and here, then you'll have your positive net movement in one way or another. Yeah. Absolutely. All right, so okay, so you got, do I need energy, right? No, you don't need energy. It's called passive if you're just following diffusion. In this case right here. But when you're needing to use energy, or we call it active transport, that's going to be when you go against the concentration gradient. When you go from low to high concentration. If we go back to the example of your lab last week, let's say during the cleanup process, I was really mean and I said, hey guys, to clean up, I want you guys to get all of your methylene blue back to the center of your petri dish. Is that going to be easy? No. You'd have to put a lot of effort and energy if you somehow had to gather all of the methylene blue that was spread out and put it back into a concentrated center. Right? So anytime you go against the concentration gradient, that's always going to require energy. So in this example, if you took these few molecules on the inside and needed to pump them out, right, from low concentration to high, that is going to require energy. And when we require energy, we call it active transport. You have to actively transport the molecules. They're not going to go on their own that way. And our main source of energy, anyone want to guess?
ATP.
Yeah, ATP. Yeah, so ATP will be used in most cases to do this active transport. Right? So again, to answer the question, is energy needed, really take a look and see where are my concentrations. If I'm going from high to low, no energy needed, passive. But if I'm going against from low to high, then yes, you do need energy. It's active. The energy normally comes in the form of heat. Okay, more questions on this? All right. Okay, so we've already quickly reviewed diffusion, but I'm going to throw that up there just to be certain. Remember, it's the movement of molecules from an area of high to low concentration. And we say that this is moving down the concentration gradient. A good classic example that we talked about last week would be dropping a cube of sugar in water. Right? You drop the cube of sugar in, it's highly concentrated sugar in the cube, low or no sugar elsewhere. Given time, what happens to that sugar cube?
out.
Yeah, it dissolves or it diffuses, so given time, right? The sugar moves from the high concentration to the low, so it moves out, and it keeps diffusing until it's evenly spaced throughout the glass. And at this point, we're finished with diffusion. We have an even spread. The concentration is even across. Okay? And again, our classic examples from lab last week when you guys did the methylene blue and the potassium permanganate, you put them in the center or roughly in the center, and given time, you saw the molecules move from the high concentration in the center where you dropped it out to the low, and then that caused the molecules to spread throughout the water in the petri dish. Okay. All right, do you guys recall there's two things you need to double check to make sure diffusion will occur? What are the two conditions you absolutely need for diffusion to occur?
Permeability.
Okay, one, concentration gradient, right? You have to see a high and a low. And what was the second thing?
Permeability.
Right, permeability. Remember the crystals aren't permeable in air, so if you were just to have potassium permanganate or methylene blue in the air, there's no diffusion because it's not permeable in air. You had to place it in water because that was permeable. So double check, make sure you have your two conditions, right? The concentration gradient, the high to low, and the permeability. Now we're going to start to migrate the concept of diffusion into the cell. So we're going to start first with an example your book gives. It's talking about a dialysis membrane. It's supposed to mimic a cell membrane. So you can kind of imagine that inside here is the cell and then here is the surrounding outside. And we're going to look at three different types of molecules in this set. The first molecule we're going to look at is this big purple protein. So take a look at these big purple protein balls. Right? And again, this is supposed to be mimicking inside the cell, and then this is outside. Where are all the purple proteins located?
Inside the ball.
Okay, inside. So we have a high concentration of the purple proteins inside, low or no concentration outside. So based on concentration gradient, where should those purple proteins move?
Outside.
They should move out, right? But notice, they're not moving out. None of them are moving out. Why are they not moving out?
They don't fit.
What do we not have?
Permeability.
Yeah, we don't have the permeability. In other words, we don't have a way to get the molecules across the membrane. In fact, these proteins are too big, there's no channels or there's no means to transport them across the membrane. So they're stuck inside because we don't have the permeability we need. Okay, now let's take a look at these smaller molecules, the small yellow ones. Now take a look again. Where are most of your small yellow molecules located?
Inside.
Inside. So based on concentration gradient, where are they going to want to go?
Outside.
Yeah, outside. And notice, because these molecules are smaller, the membrane is permeable. They can go through the membrane. And notice you see some of the little yellow molecules then moving out. So they're able to diffuse from high to low concentration, from inside to out. And then finally, let's take a look at these small little green glucose molecules. So the glucose molecules, if you take a look, they're actually about evenly concentrated inside and out. So there's no necessarily a concentration gradient. There's no high and low. It's just even throughout. Does that mean the green molecules are not moving at all? No. How are they moving?
In and out.
Yeah, both in and out, right? So you'll notice these guys are both going in and out at an even rate. And that would be, for example, like your red blood cells in the isotonic solution, right? Like one of the questions I asked you yesterday, right? How do red blood cells move in an isotonic solution? Evenly, they move in and out evenly.
So what role does the medium play in permeability? Because in permeability, we need, obviously it needs to be big enough for something to go through, but doesn't medium play a role as well? Because the permanganate wouldn't have diffused if it was in air, like you said.
Right, exactly. And so that the question that Rajan asked is, what is the role that the medium plays in permeability? The medium is like, what is the solution or what is this in? And that's why I was saying that it's mostly a water environment, right, in our body. So when we're talking about molecules moving, we're saying, hey, the medium is actually mostly water.
Okay.
And water is a very permeable substance. So most of the things we're talking about are moving in water, and that medium is permeable to all the molecules we're talking about. Yeah. Great question. Yeah, you can imagine, right? If this were just a bag sitting outside in air, you wouldn't get the same movement, right?
Right.
That's why we're talking about, hey, water inside and out. That way we don't have to worry about that medium so much.
So it always has to be in the same, for diffusion to occur, inside and outside, the medium needs to always be the
Oh,
same?
that's a great question. Does the medium always have to be the same inside and out for
Right.
diffusion to occur? Actually, no. Think about how your lungs interface with your blood vessels.
Mhm.
That's the air and a water medium on either side. But we can still get diffusion between the two because gases can be permeable both in air and in water. Right? So you just have to make sure your molecules are permeable on both sides.
Like a gas reaction. and stuff.
Right.
Okay.
But you don't have to have matching medium. Good question. All right. Okay. So, oh yeah, Daryl.
So, let's say you have a substance that got a really loud smell to it.
Okay.
Is the molecules in that permeable from solid to gas?
Yeah, great question. So let's say like you have a really potent smelling molecule, right? You put it in, it's here in the air, and then it starts to
Fill up
obviously
the room.
diffuse and fill up the room. Is that molecule permeable from a solid to the air or the gas? Yes. Yeah, so it's in a solid form, and then it can go from the solid form, and then it's permeable through the air of the gas form. Yeah. All right. So let's now get more specific. Let's actually address the phospholipid bilayer. So if we want to have diffusion through the plasma membrane, we have to review what the plasma membrane is made up of. Remember, the majority of the plasma membrane is this phospholipid bilayer. Right? And do you guys remember that the phospholipid bilayer has these little heads, which are the balls, right? And then they have these little tails, which are on the inside. Do you guys remember what hydrophilic means?
Likes water.
Right? They like water, right? So the heads, remember, are facing the inside and outside of the cells, which is watery. And then remember the tails were hydrophobic. What did that mean? They didn't like water, right? So the tails are in the middle. So keep this in mind because when a molecule has to go through the phospholipid bilayer, it has to go through a reasonable hydrophobic area in the middle. The whole middle of your bilayer is hydrophobic. In other words, doesn't like water, it's not water soluble. So keep that in mind because that's going to determine what can go through. Can it go through that hydrophobic tail layer? So what can go through? First of all, based on size, the molecule has to be relatively small. If it's a large molecule, it won't be able to squeeze through the phospholipid bilayer. That's the first thing, small. The molecule has to also be non-polar. You guys remember what non-polar means?
It doesn't have a charge on either side.
Yes, shared equally, right? So think about the charges, right? Polar means it has poles or it has charges, positive, negative. Non-polar, right? The charges are spread evenly, so it's basically not charged, right? A non-charged molecule. And the reason why, again, the non-polar can go through, if it's not charged or non-polar, that goes really well with the non-polar tails in the middle. So the tails, which we say are hydrophobic, they hate water, the tails are also non-polar. And non-polar substances like other non-polar substances. So that allows them to go through that tail layer in the middle, the thick middle layer. Another way to describe nonpolar, if there's no charges, then we say it's lipid soluble. Lipid soluble means it can dissolve or it can mix with lipids. And remember, this is a phospholipid bilayer. So if it's lipid soluble, that means yes, it likes lipids, it can go through lipids. And again, the molecules that are lipid soluble are the ones that tend to be nonpolar with no charges, because lipids also have, for the most part, no charges. There is one exception, and that's why I threw this up here, is water. Water is small, but you might remember water is a polar molecule, right? Remember the oxygen pulls on the electrons more, so it has slightly negative and positive charges, but this is an exception that is able to still go through the phospholipid bilayer because it's small enough. Other great examples that fit the nonpolar, small lipid soluble are actually certain gases, like your oxygen and your carbon dioxide gases, right, your respiratory gases. It's fantastic that they can go straight through the phospholipid bilayer because that makes for more efficient gas exchange. If you look at O2 and CO2, they're both relatively small molecules, they're nonpolar, they can squeeze right through that phospholipid bilayer. Okay, so now if we can understand that these molecules can go through the phospholipid bilayer, the next slide is going to address what cannot go through. So for example, if you know small molecules can go through, what cannot go through?
Large.
Yeah, so the first thing is large. If the molecule is too large, it cannot squeeze through the phospholipid bilayer. All right, if nonpolar molecules can make it, what cannot make it? Yeah, polar are charged molecules, right? Remember, if it's a polar, it has poles, it has charges. So for example, ion, right? So any element with a positive or negative charge can't go directly through the phospholipid bilayer. But there is a solution, and we actually already talked about it. If the molecule is large and or charged, it doesn't mean it's hopeless. It can still get through the plasma membrane, but instead it just has to use a channel. Right? You have to have a protein, such as this one here in yellow, to help these large charged molecules go across. And these channels usually come with gates. Gates just means that you can open and close them, so you can control when these molecules go across. Okay, question? Yes?
Is this active or passive?
Oh, that's a great question. Is this active or passive? To answer that question, what do you guys have to ask? You want is it active or passive? What do you need to look for?
Energy.
Right? Is it using energy or is it not? Or in other words, are we going with the fusion or are we going against? So this can come in active and passive form. It really just depends if you're using the energy or not, if you're using the fusion or not.
So is active or passive?
What?
So is this
I said it could be both,
both? Okay.
right? You just have to look at the situation and then ask yourself, okay, these molecules that are moving across, are they going down their concentration gradient with diffusion? If that's the case, then it's passive. Or is it using energy and going against? Then it's active.
Okay.
Yeah. So it can go either way. Okay. If you look for like this particular scenario, right? Let's take this as an example. In this particular scenario, can you see a high and low concentration gradient? Yeah. And this side, what's on the right? High. What's on the left? Low. So if the molecules were moving in this direction from high to low, which one would that be? Active or passive? Passive. Yeah. So if they were moving this way, it would be passive. But if I told you the molecules were moving the opposite way, then what would that be? Active. Yeah. So again, it depends on the scenario. You have to check out that scenario to see if it's active or passive. Okay. All right. Now here's another quick review from last week. Remember what can affect the rate of diffusion? Remember the first is concentration. How does concentration gradient play a role? What happens if you have a larger concentration gradient?
It moves out.
What happens to diffusion? Faster or slower? You have a larger concentration gradient. Faster, right? A lot of you guys explained, for example, with the methylene blue, you used a higher concentration, which meant a higher gradient, which meant faster diffusion. What about temperature? How does that affect diffusion? The hotter, right? The faster the diffusion goes, right? That was another concept on the exam, right? I asked, hey, if you put these molecules in 40 degrees Celsius versus 27, right? Which one's going to diffuse faster? The hotter water, the hotter temperature, the faster the diffusion. And then molecule size. How does molecule size affect diffusion?
Smaller goes faster.
Smaller molecules diffuse faster, right? If everything else is constant, the smaller molecule will go faster. And then the last thing is surface area. And this is what we discussed, for example, the methylene blue was in a finer powder form, right? That finer powder, we said, had more surface area. If it has more surface area, how does that affect the rate of diffusion?
So
Sure.
more surface area, faster diffusion.
Faster diffusion again, right? The more surface area, the more contact you have, the more contact, the more you can move, the faster you can diffuse. So that would be, again, the methylene powder being finer, smaller, more surface area, faster diffusion. Okay? You can just keep that in mind. You can always kind of go back to your concrete examples of lab to help you understand these concepts of how diffusion rate can vary. Okay. All right. Then, after diffusion, we can get a little more specific and talk about osmosis. Remember, osmosis is just simply the diffusion of water. So in this particular case, you guys were focusing on the movement of water from an area of higher to lower water concentration. So your book uses this figure as kind of a generic experiment example in a petri dish. So we have a beaker with a selectively permeable membrane. This membrane is only permeable to water. And we have two sides, A and B. The little blue molecules represent water. The purple molecules represent protein. So if you take a look, which side has more protein, A or B? A. Okay, so if A has more proteins, or we say more solutes, more proteins or more solutes means less water. So side A has less water or a lower water concentration. Side B on the right, notice has less proteins or less solutes, meaning that it has more water instead. So side B has the higher concentration of water. Side A has the lower concentration of water. So based on osmosis, where's water going to move in this scenario?
A to A.
From B to A, right? Or in this case, from right to left. So the water's going to move over in that direction, high to low. And the reason why the water can move over is because, remember, this membrane is selectively permeable. It allows only water to go across. So if you were to zoom in on this membrane, you'll see that all the little blue water molecules can fit across the little hole. But notice the big purple proteins cannot move, right? The membrane is not permeable to the protein. And in fact, a lot of times, if you want to just selectively move water across a membrane, we use these special little channels called aquaporin. And fortunately, this is named pretty appropriately because what does aqua mean? That just means water, and pore, a pore is a hole. So aquaporin is just a hole for the water. So we'll see that kind of later on as we move throughout the entire body. There's a lot of special just aquaporins that we can just transfer water through. So given enough time, and eventually the water moves over to the left side and you'll start to see the water build up on the left. Right? Again, until it's evenly spaced out. Right? Okay. So let's work our way closer to an actual body cell. So in this particular scenario, we have a beaker, and in the middle we have what's called a semi-permeable membrane sac, or in other words, just think of it as mimicking a cell. And the little blue in there represents sucrose, or a sugar. So notice there's a high concentration of the sucrose inside this little sac. Again, if there's a high concentration of sugar, or the solutes, that means there's low water concentration. So inside the sac is low amounts of water. Outside the sac are high amounts of water. So what do you notice about the direction of water movement here?
Into the sac.
Yeah. The water is all moving inside. Right? So again, from high to low.
The
Okay.
question, so, but wouldn't the capability of the cell to allow a certain amount of water come in eventually restrict? Unless
Yes.
unless it goes under lysis, right?
Exactly. So yeah, so in this case, right, the water will keep going in
Right.
until either one of two things happen. One, the little sac or cell could burst, right? We talked about lysine, right? Or two, maybe this membrane is strong enough and it'll actually restrict more water coming in. Right?
And then the concentration would still be different though.
The concentration might still be different. And
Right.
that is actually talking about, I'll bring it up next,
Okay.
that's something called osmotic pressure.
Oh, okay, all right.
Yeah. Okay. So let's go back and review what we did last week, one of the scenarios of water poisoning. So try to recall what happens in water poisoning. In other words, when you put your red blood cell in a hypotonic solution. Did 1, the red blood cells crenate or shrink? 2, did the red blood cells lyse or burst? Or 3, sorry, it got cut off, were the red blood cells not affected? They're the same size. What do you think? Was it 1, 2, or 3 for water poisoning? Alright. Okay, good. I've seen majority 2s and some 1s, so you guys know something's going to happen with the red blood cell. So, 1 or 2, let's take a look at the hypotonic solution. Right? Hypotonic solution, remember, was similar to like distilled water, or when we say water poisoning, too much pure water. So in this scenario, in hypotonic solution, outside the cell is basically pure water, and then here it's inside the cell. So we have a high concentration outside the cell, low concentration of water inside the cell. Where does the water move?
Into the cell.
Into the cell. If more water moves into the cell starts to swell. Eventually, the cell will lyse.
So when you say pure water, that means it's only H2O present?
Yeah. Yep. So if you're like in pure, you put a red blood cell in pure water, distilled water like we did last week, right? Only H2O present, then the water is going to move from high to low or from out to in.
So what other molecules is in the water we drink?
Yeah, so good. What other molecules are in the water that we drink? It really depends if you're drinking pure water or not, right? If you're drinking pure distilled, it's just pure water. But otherwise, usually what's in the water you drink, for example, in the faucet water you drink, there's going to be little traces of chloride. Sometimes there's actually some small traces of sodium and potassium in the water. So there's always some slight solutes dissolved in the water. When you buy, for example, like Evian or Dasani, all of those waters will have some trace little ions in them.
So what about spring water?
Same with spring water. Yep, because they're running, spring water comes, you think of spring water is coming from a mountain, right?
Right.
They're gonna purify it and whatnot, but there's still gonna be trace ions in that water, which is why it's not actually distilled water, right? So the only true just pure water is distilled water, and you really don't see that except for like when we run our experiments, especially in chemistry, a lot of times.
It's not healthy to drink distilled water, right?
Is it what?
It's not healthy to drink.
It's not healthy to drink large amounts of just pure distilled water, right? Because then we have this scenario. But remember, I said to get to that point where it's that unhealthy, where it bursts your cells, you have to be drinking so much within a small amount of time for it to actually burst your cells.
So there's actually people that drink distilled water on the regular?
For the most part, you don't. No, because distilled water, again, you don't, any water you're going to buy at the store is not distilled for the most part. Yep. Right, good question. All right, so again, high to low, water moves into the cell, cell swells and can lyse. Now, this doesn't happen with all cells. Yes, it happens with our red blood cells, right? But a lot of cells have what we call osmotic pressure. That's the pressure based off of the cell membrane pushing in, and it's the pressure that actually stops osmosis at some point. And the hope is that this pressure would be enough to stop osmosis before the cell actually bursts. Unfortunately, red blood cells, yes, they can swell to a certain extent because they have this pressure, but if they start to swell too much, they're going to be susceptible to bursting. That's why we always want to remember keep our red blood cells in the isotonic solution. Right? You guys remember what percentage saline is isotonic?
0.9.
0.9%, right? That's what they use on the saline bags. You want to make sure you're in isotonic so you don't have the changing of the red blood cell size. Because again, if you put that red blood cell in too much water, in a hypotonic solution, again, the osmotic pressure of the red blood cell can only withstand so much water filling up before it eventually bursts. Plant cells, on the other hand, have a much stronger osmotic pressure. You might remember from basic bio that plant cell walls are very rigid. They're a lot tougher than animal cell walls. So that's why it's actually a lot harder for a plant to get water poisoning, right? You can overwater plants. They might not look so great, but for the most part, they're not going to kill over as fast as, for example, a red blood cell is going to burst, because you can load up a plant cell with water, but because the wall has that great osmotic pressure, it basically keeps the force pushing inward so it doesn't burst open. Okay. All right. So then just to review, we've been looking at these kind of different solutions. We talked about hypotonic with the swelling of the red blood cell. We talked about the ideal, which is isotonic, where your red blood cells keep the same shape because we have even concentrations of water inside and out. And then, so in this case, right, water goes in and out evenly. And then the last solution, remember, is hypertonic. And for the red blood cells, this is anything above 0.9% saline solution, right? 3%, 10%. In this case, you start getting too many solutes outside the red blood cell. For example, too salty, right? Or if you're dehydrated. And in that case, remember, the water goes outside of the cell, causing your cell to shrink or cremate.
Might be slightly off topic, but does sea animals have a stronger osmotic pressure than land animals?
Oh yeah, that's a great question. Do sea animals have a higher osmotic pressure in their cell wall? Because you might be thinking, oh, they have to withstand a little bit more like differences in solution. As far as I know, their cell wall or their cell membranes are the same as ours. If you think about it, the sea animals in this case would be, if anything, more susceptible to this solution, right? The salty solution, and so they're not worried so much about their cells lysing and bursting. They would probably have to be more concerned about the dehydration aspect or the shrinking of the cell. All right, other questions?
Quick question.
Yeah.
Sorry about the hypertonic solution. So what happens, like usually, so I get it for the isotonic, it leaves the cell, for the hypertonic it goes in the cell. But for the hypertonic, will it be just dehydration?
For this one? Yeah, so again, this would be dehydration, right? You didn't drink enough water.
Okay.
So you have too much, for instance, not enough water outside your red blood cell. So remember, that makes the water concentration low outside, but high inside, which means the water is going to go where? Out, right? So if the water leaves out, remember your red blood cell then starts to shrink, right? Because the water goes out of the red blood cell. And that was cremate, or cremation. All right. Okay, let's now, that we kind of reviewed diffusion and osmosis, let's go back and take a more specific look at carrier-mediated transport, because this is going to become really critical in getting some essential things across our cell membrane. For example, the best example is glucose, right? Remind me again, why do we need glucose to enter the cell?
Cell energy.
To make energy, right, through cell respiration for glycolysis. So the way glucose enters the cell is through the cell-mediated transport. So let's take a closer look to make sure we can do that efficiently. All right. So remember, carrier-mediated transport, the hallmark is that the protein has to physically touch the molecule, carry the molecule into the cell. So when we look at these proteins, remember, proteins can be enzymes. And what do enzymes do? What's their job? Yeah, speed up the reaction. So these carrier proteins, what they're speeding up is the movement of the molecule across the cell membrane. That's the reaction they're speeding up. So when we look at these enzymes, we notice first is that they're very specific. Remember, enzymes only work on specific substances, like salivary amylase, right? We learned that it only works on breaking down starch, right? From the exam yesterday, remember I asked you, hey, is it going to break down protein? And hopefully you guys said no, right? It's not going to break down protein because it's specific for starch. So the same thing here. These carrier proteins are very specific and they will only carry across a specific molecule. For example, if this one is working on glucose, it will not work on any other molecule. It won't even work on sucrose or fructose. It is specific for glucose. The second thing is that there's competition. There's always more than one carrier protein, right? Here we're just showing two, but they're competing for the same molecule. So there's a competition to get the molecule and bring it across. another feature is that these molecules have a saturation point. What does it mean to be saturated? Full, maximum, right? We talk about like saturated fatty acids, they're full or maximum amount of bond. You might even say like, hey, after a meal, I'm very saturated or I'm very full, right? So saturation is you're at your maximum capacity, you're completely full. Another word, it would be like, these proteins are working as fast as they can. They can't carry across anymore at any faster rate. They're working at their maximum or full capacity. But at some point, right, these proteins can become saturated or they can't work any faster. Okay.